Welcome back. Now, we will proceed to describe the chemistry of other whatever remaining B complex cofactors and study the reactions that they in which they participate along with the enzyme. Okay. So, what is left now is cobalamin, and cobalamin is the vitamin B12 is the coenzyme. Cobalamin is the vitamin is the coenzyme form from vitamin B12. Okay. Now, the structure of this coenzyme, which is cobalamin, it has got the name has cobalt, suggests that there is cobalt in it. So, this is a tetra pyrrole type of ring, and it's attached to the in a square planar fashion, coordinated to the 4 nitrogen atoms, and however cobalt is hexa coordinated. So, in the on this one, this side, in this case the top side, there is an adenine moiety, where the CH2 wedge is replaced. If you remove the wedge, so then this CH2 can be attached to the metal cobalt. That means, it is an organometallic. This is an organometallic system, because organometallic means organic, there is a carbon metal bond, and that is here organometallic system, and there is an organometallic bond between the CH2, that is the 5 prime CH2 in adenine, and then in the form of adenosine. This is because this is attached to the adenine to the sugar. So, this is adenosine. The CH2 is attached to the cobalt, that is a covalent bond, and on the other side of the hexa coordination, the other side. So, 4 sides are occupied by these nitrogens, and the other 2 sides, one is the CH2 of the adenosine, and other is this is. So, all these 5 membered rings, the nitrogen, these the pyrrole dihydro or the pyrrole moieties, they are attached to this acetic acid moiety, or forming the amide, not acetic acid. So, acid-amide moieties, but in one of these there is a methyl here, and then that the other, this is a propionic acid, CH2, CO, and then propionamide, and then you have this is your lactate, CH3 CH OH. No, this is not lactate, this is your CH2. So, this is nothing but derived from NH, CH2, CH, CH3 and OH. Okay. This is the compound. So, this is not any acid, amino acid, this is an amino alcohol. So, the amino alcohol is attached, here is the amide bond, and here it is attached to the phosphate, and then phosphate is interestingly attached to this, this is the 3 prime, that is so okay. So, 1, 2, and the 2, 1 is a N glycoside, but this is a different type of base, not encountered earlier. Okay. This has got an imidazole, and the imidazole nitrogen is directly coordinating to the cobalt. So, that is a very complicated type of structure. So, you have these pyrrole moieties, and then occupying this these 4 sides here, and then you have the methylene attached to the cobalt of adenosine, and on the other one, this is a new base, this is a bench triazole, the triazole nitrogen is coordinating to the cobalt. So, that is the structure, but we can just simplify it, that CO 3, and you can just, because this is the actual functional part CH2, and you can write AD. So, adenosine CH2 attached to cobalt, and we are not concerned about whatever the other things are, okay, because this will be sufficient in order to explain the reactions. Now, vitamin B12 consists of 4 pyrrole rings, it says, they are not really perfect pyrroles because they are substituted pyrroles and they are in different oxidation level. And then cobalamin participates in the synthesis of acetylcholine, that is very important. Acetylcholine is another neurotransmitter. We talked about some neurotransmitters in some of the sessions of PLP dependent enzymes. Deficiency of vitamin B12 causes anemia. Okay. So, let us see what type of reaction it catalyzes. There are, I just pointed out two reactions here. One is methyl malonyl CoA, that what we have encountered just in the previous session. So, this methyl malonyl CoA can undergo rearrangement. See, this participates in reactions which are isomerization reactions. Okay. So, this carboxy can migrate to the terminal carbon. So, this has migrated to the terminal carbon and one of the hydrogen migrates here. So, in the resulting is succinyl CoA. So, you have started with methyl malonyl CoA and by this is also mutation reaction because this is change of the constitution of the molecule. So, if you change the constitution, those type of enzymes are collectively called mutase, but they are basically isomerase. So, through this isomerization you get succinyl CoA because their formula remains the same, molecular formula remains the same, only their constitution remains different, becomes different, that is a mutase. You can have glutamate mutase. What happens here? This CO2, this is beta methyl aspartate, you have a methyl here and this is aspartic acid up to this point and then you have additionally a methyl. So, beta methyl aspartate. See, if the carboxy is migrating to this carbon and the hydrogen goes to this carbon, then what happens? You have this glutamate. So, beta methyl aspartate can be converted into glutamate. The name of the enzymes are glutamate mutase and this is methyl malonyl CoA mutase. Okay. Methyl malonyl CoA mutase. Of course, this can be called if it is glutamate mutase, you can call it actually beta methyl aspartate mutase because you are doing mutation on this and going to that. I think that should be the case, but in the books that is what is written, but my feeling is that it is better that you call it beta methyl aspartate mutase, which gives you glutamate. This is methyl malonyl CoA mutase, which gives you succinyl CoA. But the thing is that these reactions are reversible. So, for the reverse reactions you can call this as glutamate mutase and you can call this as succinyl CoA mutase. Okay. You understand the point, no? Because what type of which direction you are following, accordingly you can name the enzyme. Okay. Now, let us see how these mutations are done. What happens here? I told you this is adenine. The simplest way of writing this vitamin B12 chemistry is that this is the cobalt and there are other ligands here. So, first, because of this bond will be very reactive bond, very unstable. So, it can go into it can undergo homolytic fission and forming the adenine CH2 radical, and obviously, the cobalt will now get back its electron that is as donated here. So, cobalt will become cobalt 2. So, this is the scenario. Now, you have, suppose I do this reaction plus CH3 CH COO minus and then CO SCOA, okay, methyl malonyl CoA, and then what happens? This takes up the hydrogen. Let me just see which hydrogen it takes. Methyl malonyl CoA, that is not given here. So, obviously, it should abstract hydrogen from this. First hydrogen to be picked up should be this, because that gives a stabilized radical CH C dot, not CH, CO SCOA, and this will be COO, suppose H, better write now H. And then if you migrate one hydrogen from here, so you get dot here and then CH, the hydrogen is there, thou CO2H, and you have C SCOA, then the CO2H moves here and you get CH dot COSCOA. Okay. And then all along this one, so this one will become AD. So, first hydrogen abstraction, it becomes AdCH3, because it is this dot which abstracts the hydrogen from here. So, it becomes CH dot, and then finally, the adenosine CH3 was waiting, it is holding the hydrogen till the proper radical is formed here, the CO2H has migrated. So, now this will become CH2 CO2H, and that will lead to what is called succinyl coenzyme A, and the adenine becomes AD CH2 dot, and then it can add to the cobalt which was waiting to be combined again, and it forms the vitamin, the coenzyme form of vitamin B12. So, again just to summarize this process, what happens here, because this is a very reactive bond, organometallic carbon metal bond. So, that can homolytically dissociate. So, resulting in a radical at the adenosyl part, and the cobalt goes to cobalt 2, and then this abstracts hydrogen first here. So, it gives a dot, and then the hydrogen migrates from here and goes to there. So, CO2H then migrates, resulting in a radical producing here, the CO2H migrates here, again producing the radical at this position, and then this abstracts the hydrogen from the AdCH3, adenosine CH3, resulting in formation again back to AdCH2, and that can combine with the cobalt 2 complex to make the coenzyme of vitamin B12. Sorry, vitamin B12. Okay. So, that is the mechanism. The other mechanism that is possible is that this initially abstracts hydrogen from here, CO2H migrates there and it forms the radical here, and then that abstracts the hydrogen from the AdCH3. That is also possible. Okay. Here are again other reactions mechanisms are shown here. One reaction is that if you take this propane 1,2-diol, it has been found that propane 1,2-diol, this becomes, the ultimate product is ArCH2CHO, methyl, that means propionaldehyde. So, propane 1,2-diol goes to propanal. That means, to have propanal, so you have to move this OH to this carbon, so that this carbon bears 2 OHs, and then one water molecule will be lost, resulting in the formation of the aldehyde. Okay. So, basically what happens here, it abstracts the hydrogen from here, forming a radical at this position, then OH migrates from here to there, forming a radical here, and then this abstracts the hydrogen from the methyl of the adenosyl part, and then you can again form the vitamin, the coenzyme form of vitamin B12 complex. So, this is a unique mechanism. Up till now we have not encountered any radical type of reaction. This is this cobalamin chemistry or the vitamin B12 chemistry is the reactions are done via radical intermediates. Okay. And people have used this vitamin B12 coenzyme form for carrying out, for generating radicals and doing radical reactions. So, that is a separate part of organic chemistry, which where radical chemistry, synthetic radical chemistry, but the starting point is the coenzyme form of vitamin B12. Okay. So, now I think FAD we have already covered. FAD FMN is very similar to what FAD does. This mechanism we have shown that this is the oxidized form of, this is FAD, and then FAD goes to always FADH2, and then FADH2 can be converted back to FAD by NAD plus or NADP plus. So, let us go to the, because we have covered this, quickly go to the NAD plus and NADP plus. So, what are these? These are very, again, structure is quite complicated, but the vitamin form is actually nicotinic acid. Nicotinic acid is pyridine 3 carboxylic acid. So, the coenzyme, this is the coenzyme form. So, nicotinic acid is converted to nicotinamide, and then this is the ribose, then you have this bisphosphate, and you have this or the pyrophosphate, and then again you have ribose, and then that is attached to adenine. So, this is called nicotinamide adenine dinucleotide, okay. That is NAD, and sometimes this X may be a phosphate group, and that that gives you nicotinamide adenine dinucleotide phosphate. The phosphate is basically including the oxygen. So, if this is fully the phosphate group, then that is called the NADP plus. Some reactions are catalyzed by NAD plus, some are NADP plus, depending on the enzyme. Okay. Now, what is the role of NAD plus? That if a substrate is reduced, if a substrate is in the reduced form like FADH2, then you have to have NAD plus, the substrate is oxidized and NAD plus is reduced to NADH, or it could be NADP plus that works on the substrate, reduced substrate, and then in presence of the enzyme you get the oxidized substrate and NADPH plus 1 proton. Okay. So, there are many reactions that are shown here. One important reaction in glycolytic cycle is this one, that D-glyceraldehyde 3 phosphate in presence of NAD plus and a phosphate, it goes to D13 diphosphoglycerate. So, there is an oxidation, the aldehyde goes to the carboxylic acid, and that is converted into the phosphate. Okay. That is converted into the phosphate. So, basically you have to oxidize this, and the oxidizing agent is NAD plus, but this, remember, there is an enzyme requirement for this. Means if in a test tube, if you take this and add NAD plus and phosphate, some amount of this you can get, but that will be extremely slow. It is the enzyme which brings all these three components together and does the reaction. There are many such examples which are there. The question is how NAD plus works. Okay. Now, NAD plus, again it is a complicated structure, but the functional part is only this one, this nicotinamide. So, we can write NAD plus always in the abbreviated form R, and the nitrogen is positively charged. As nitrogen is positively charged, like pyridoxal phosphate, it becomes an electron sink, and this fourth carbon is very vulnerable because of the pull, electron pull by the nitrogen. So, what happens if you take an alcohol, then the hydrogen can migrate as hydride, as a nucleophile, attacks this carbon, and so there are two hydrogens now here, and in the process this alcohol becomes aldehyde. So, there are many such reactions. Now, alcohol dehydrogenase. Alcohol dehydrogenase means it is in presence of NAD plus or NADP plus that you get from the alcohol, you get to the aldehyde. And where is the hydrogen gone? This hydrogen from the alcohol part goes to the this is what is NADH or NADPH, depending on what is in there R, NADPH. Okay. And for the reverse reaction, see NAD plus NADH, this type of one is oxidizing agent, another is reducing agent. So, whenever you require to oxidize, the hydride is added to NAD plus. If you want to reduce something aldehyde to an alcohol, that is also possible, these are reversible reactions. Then you take, but that is done by another enzyme, then you take this NADH and then transfer the hydride from the NADH to the aldehyde and resulting in the formation of the alcohol, and NADH goes to NAD plus. Now, all these migrations of hydrogen, hydride transfer, sorry, this is not NADH, this is NAD plus. Okay. Now, all these migrations have been demonstrated that this is the hydrogen from the NADH that migrates and attacks the carbonyl carbon, or the reverse, the alcohol hydrogen, the carbonyl carbon hydrogen, if you make a deuterium here, and that is what is transferred to this aromatic ring. And not only that, you have a stereochemical part here, and that is that these two hydrogens are basically enantiotopic because of the you have different chiral groups here. Okay. But truly speaking, if you you can make these as actually they are not enantiotopic, because if you have a chiral group here in the form of adenosine and those things. So, actually these are diastereotopic, but one will be you can assign them as pro R and pro S, pro R and pro S. That means, they are stereochemically they are basically different hydrogens. So, in one case it is the beta hydrogen and this is the alpha hydrogen, and you know how to know the pro R pro S. Again just remind you, if you most of you must be knowing, but I just repeat that this hydrogen, in order to know whether this is pro R or pro S, you give preference in the priority order to this hydrogen over the alpha hydrogen. So, this will be your 1 in preference, this will be your this will be your 1, not that one, this will be your 2 and that will be your 3. So, that is 1 to 3. So, this is pro R and that is your pro S. You do not have to do, if one is pro R, the other will be pro S. So, all these reactions, either it could be the pro R hydrogen going to attack the carbonyl carbon, or it will be the, here also there will be pro R pro S hydrogen. So, one of these hydrogens only will be transferred. So, if I say that in this reaction, suppose I have a deuterium here and hydrogen here, and I do this NADH mediated reaction, and I have an aldehyde, and I ask you to what is the product if pro S is migrates. So, what will be the product? So, product will be RCH2OH, because the pro S hydrogen is this one. So, that is what is the enzyme likes to migrate. So, this hydrogen will be taken up and you will get RCH2OH. If it is, this is pro S, if there is pro R migration, then you will get RCH2OH. But remember, now you are making a chiral center. So, that will also be, deuterium will be delivered in a particular way, either from the ray phase, because you are now adding to the carbonyl. So, it will be either from the ray phase or from the Shy phase. So, there are stereochemical issues involved in NAD mediated reactions. Okay. It is basically the hydrogen that is involved here. Now, the last one, there is one slide here which says that how the carbonyl is, this is basically the glyceraldehyde 3 phosphate. I told you glyceraldehyde 3 phosphate goes to this is the phosphate, glyceraldehyde 3 phosphate goes to 1,3-CO2 phosphate, and this is CHOH and this is CH2OP. So, 1,3-bisphosphate, but that is glycerate, this is glyceraldehyde. Okay. So, what is the possible mechanism? Gap is this glyceraldehyde 3 phosphate. So, what happens if you can write this as R, the whole thing. So, it will be RCHO. Initially First there is a cysteine moiety that that delivers the hydrogen here, the hydride. Okay. So, the hydride goes here, sorry, the base takes the hydrogen and sulfur attacks the carbonyl. I am sorry, base takes the hydrogen and this attacks the carbonyl and this becomes O minus, the tetrahedral intermediate that comes back, and now it is like a very similar to Cannizzaro reaction. This O minus comes back and hydride now is going to NAD plus. So, NAD plus becomes NADH. Okay. So, this becomes SCOR, so thioester, and this will be NAD, this will be NADH. Okay. Now, this NADH, what is here? This is the phosphate, monophosphate. So, the monophosphate now attacks this one and basically it releases the again breaks the carbon sulfur bond and it becomes the cysteine. The cysteine, the sulfur takes the hydrogen from the base, original base which had abstracted the hydrogen. So, now this becomes what is this is R, then CO, then that is the phosphate. So, R, CO and the phosphate, that what we are talking about, that this is the R and then CO and then phosphate. Okay. So, that is the this, because this is a very important reaction in the glycolytic cycle. So, we thought that we should study it. Okay. Again just a brief summary. First, see, this is the enzyme which is the does this oxidative phosphorylation. So, the gap is the substrate, that means the glyceraldehyde 3 phosphate that goes and binds, then the cysteine attacks as a nucleophile, the base abstracts the hydrogen, the cysteine S attacks the carbonyl forming a tetrahedral intermediate, then the O minus comes back, and in a very similar to Cannizzaro reaction the hydride is transferred to NADH. Then the NADH, then the phosphate, now phosphate attacks the carbonyl and breaks the carbon sulfur bond, that is the, and that takes up the hydrogen. So, it becomes the cysteine NAD plus and then actually this one will be initially it will be NADH, but that will be later on oxidized to the NAD plus, and then SH you get your product when the after the expulsion of the carbon sulfur bond, you get what is the phosphoglycerate, 1,3-phosphoglycerate. Now, the last one is vitamin C. This is the last of the coenzymes that we want to discuss. Okay. The vitamin B group is now complete. We have discussed vitamin B12, that is cyanocobalamin. We have discussed the NAD plus, NADH, NADP plus, NADPH, and we have discussed the other vitamins like the vitamin B 6 PLP, then the PLP is the coenzyme, then folic acid, then we have discussed biotin, we have discussed the thiamine, all these have been discussed. So, we have covered most of it. Now, only vitamin C is remaining, and vitamin C, we know that this is also a very important enzyme, coenzyme, and that is called ascorbic acid. Okay. The ascorbic acid is, the structure is given here. Okay. Now, this is what is called the reduced form of ascorbic acid. Now, ascorbic acid is a very good antioxidant. It scavenges out the harmful radicals that are produced inside the body. I told you time and again that it all starts with the oxygen going into the peroxo, into the superoxide. First it abstracts one electron, takes one electron, goes to the superoxide, then to the peroxide, and that can go to the hydroxyl radical. So, all these radicals are extremely damaging. So, the vitamin C can take care of these radicals, along with I told you vitamin E. Vitamin E is also an antioxidant, vitamin A is also an antioxidant, but vitamin C is extremely powerful antioxidant. However, the reaction, so as we are discussing coenzyme activity, so we will see what type of reactions it is involved in. Okay. So, it has got hydroxylation, it takes part in reactions or it helps reactions which does hydroxylation, and it also has antioxidant activity. Okay. So, what are these hydroxylation reactions? These hydroxylation reactions, like there is one muscle building compound which is called carnitine, and tyrosine also is formed from phenylalanine, because you need one hydroxy group at the para position of the aromatic ring, or you can also they are involved in hydroxylation reactions are also in case of neurotransmitters like epinephrine. We will talk about that, epinephrine or noradrenaline, adrenaline, all those things. Okay. Antioxidant activity is basically it reacts and removes active oxygen species, that means the reactive oxygen species, as I said, peroxide, superoxide and hydroxide radical. Okay. Another important thing is that it can chelate to ferrous ion to make it more soluble, because we need iron, we need iron to produce our red blood cells, and it helps to chelate the with the ferrous and then form the ferrous more soluble in the aqueous medium. Now, the reaction we are going to talk about is this one, that you know that our muscles are made up of proteins which are called collagens, and collagens are present as triple helix. So, that triple helix means it gives lot of strength. Okay. Now, we know the helix are basically formed due to hydrogen bond between the backbone, this amide carbonyl and the nitrogen NH. So, they interact, means they form hydrogen bonds and give stability to the helix. So, in order to form the triple helix, what has been found that the proline, that is the only secondary amino acid, that has to be converted into 4-hydroxyproline, 1, 2, 3, 4. So, there should be a hydroxy group here. So, this hydroxylation requires an enzyme called prolyl hydroxylase or proline hydroxylase. This is also abbreviated as PHD. Very important enzyme, because this is very important, because that hydroxylation, this 4-hydroxyproline is what is present in collagen. That means, in collagen the proline, which is the natural protein amino acids, they are further hydroxylated to form the 4-hydroxyproline. That is what is present in the peptide in the amino acids making this this polypeptide. Okay. And these hydroxyl groups gives you extra stability by forming other type of participating in hydrogen bond formation. Okay, that gives us the strength. Similarly, there is the lysine moiety, that also can undergo hydroxylation, but that will be lysine hydroxylase, lysyl or lysine hydroxylase. Now, this enzyme requires, now we for the first time we will introduce a metal ion to catalyze a reaction, the metal ion as a cofactor. We know that metal ion also are very important in some of the enzymatic reactions, and different metal ions, zinc, iron, cobalt we have already encountered, of course cobalt we have encountered. Now, we are encountering the iron, okay, the ferrous as a cofactor. So, what is the role of ascorbic acid here? So, we said that prolyl hydroxylase in a this is called post translational modification. Post translational modification means when the polypeptide is made containing it will contain all the this protein amino acids there are 20. So, it will initially contain only proline, and then once it comes out of the ribosome forms the peptide, then this enzyme prolyl hydroxylase that does what is called post translational modification, by which the proline moieties are converted into hydroxyproline. Okay. And these hydroxyprolines give you extra stability. This hydroxylation is very important, because that helps the polypeptide to form triple helix. What is the mechanism? Now, prolyl hydroxylase, as I told you, it requires a ferrous moiety, okay, iron in the ferrous oxidation state, not in the ferric. The ferrous oxidation state. Now, vitamin, to start with, I tell you the vitamin C, that means ascorbic acid, it actually this is, I told you, this is the reduced form of ascorbic acid. Now, ferrous, you know, has a very strong tendency to go into the ferric in presence of oxygen, and this hydroxylation is carried out by oxygen. And so, in presence of oxygen, there will be a strong urge of this ferrous to go into the ferric state. Vitamin C or ascorbic acid reacts if there is any ferric. So, it will now convert this ferric to the ferrous, and itself will be, that will be reduced, ferric will be reduced to ferrous, and this will be converted into the, this will be converted into your the oxidized form, and that is both are the carbonyl, both are the carbonyl groups. Okay. So, that is the oxidized form of ascorbic acid. So, ascorbic acid's duty is to keep the iron in the +2 oxidation state. We are discussing the mechanism of this hydroxylation of proline to form 4-hydroxyproline. Okay. The ascorbic acid is acting as an antioxidant. Its role is to keep the iron in the +2 state. Now, the question is how this hydroxylation is carried out by this prolyl hydroxylase or PHD. Now, this PHD enzyme, it requires iron, I already told you, ferrous, but it requires additionally another co-substrate, which is alpha ketoglutarate or 2-oxoglutarate. Okay. So, what this 2-oxoglutarate in presence of the oxygen helps the iron in this complex to form a very superoxidant, which is called ferryl oxo species. A ferryl oxo species where the iron is in the +4 state. Now, how it is formed? First, this is the resting state of the enzyme and the cofactor, that is the iron. So, it has got 3 water molecules as ligand and aspartate, histidine and histidine. Then alpha ketoglutarate, which is basically CO2H, then CH2CO, CO2H. This part you can write as R. So, this becomes RCO-CO2H. So, this acts as a bidentate ligand to form this, to expel the 2 water molecules, and itself chelates to the iron. But there is no change in oxidation level of iron here, because both the bonds are formed by electrons coming from this oxygen. So, this is the intermediate. Now, oxygen comes and iron reacts with the oxygen. Now, in this is the textbook mechanism, but there are still certain drawbacks, because the oxygen will be in the triplet state, that is the natural oxygen. So, what will happen? Iron, I can write it here, iron was in the 2 oxidation level, and you have this oxygen, that is CO, then another CO, which is coordinating to the iron. So, this is the situation. I am just disregarding other ligands. So, now oxygen is in the triplet state. So, what will happen? Now, iron will donate 1 electron here and form a covalent bond between this and that. So, this forms a covalent bond, and the resulting, the iron will be in the +3 oxidation level, and this becomes a peroxo radical, and you have iron that chelation is still there, the bidentate ligand. So, double bond O R and this was connected to the iron. Now, what will happen? Now, this radical first this cleaves, resulting in formation of, or not yet, sorry. The first is extremely sorry. The next one is this attacks here and this one of the electron comes here. And remember, this bond was formed from 2 electrons which are both coming from the oxygen. Now, one of the electron will come back here. So, it will be a lone pair, and this bond, to have this bond intact, the iron will donate 1 electron to form the covalent bond here, to complete the covalent bond here. So, that means you get Fe in the +4 oxidation state, because 1 electron earlier it was +2, then 1 electron was given to form this iron oxygen bond, and 1 electron is given to make up for the loss of 1 electron in this coordinate bond. Okay. So, iron ferryle is the iron 4, then you have oxygen CO, then this is O, and that is attached to the iron, and this is attached to. So, this is the scenario now. Now, there will be release of carbon dioxide. So, this ferryle oxo is formed. So, resulting in the formation of this is the ferryle oxo. What comes out is carbon dioxide. Now, the ferryle oxo, the oxygen abstracts, is a superoxidant, abstracts a hydrogen from here, and this becomes a radical, and this radical then adds, the oxygen adds to this radical, and one of the electron comes back to the iron. So, you get 4 hydroxy proline, and the iron is brought back to the original state. However, as I told you, in presence of oxygen, this reaction is oxygen dependent. So, also it is dependent on alpha ketoglutarate, but presence of oxygen, the iron has a great tendency to be converted to the iron 3. Ascorbic acid is basically, if there is formation of any iron 3, that will immediately be brought to the iron 2 level, and to maintain this catalytic cycle, and ascorbic acid itself will be converted into the dihydroascorbic acid, that is the oxidized form. So, that is little complicated, but I thought I should tell you this, because this will be required. These metal mediated reactions are also very important in the medicinal chemistry aspect, because many of these drugs actually coordinate to the metal ion that are absolute requirement as a cofactor in many enzymatic reactions, which are target for drug discovery program. So, that takes care of our first part of this course, the organic chemistry and biology. So, we have discussed the amino acids, the building blocks of life, molecules of life, amino acids, and then proteins. Then we have discussed carbohydrates, lipids in a very brief way, and then we have discussed the nucleic enzymes, then nucleic acids, the recombinant, the molecular biology tools, and finally we have discussed the the coenzyme chemistry, which is a vast organic chemistry, application of organic chemistry in understanding the reaction mechanism. So, from next session onwards, we will switch on to the medicinal aspects. Now, we have a very good platform on which the medicinal chemistry can be taught, because that requires organic chemistry background as well as the biochemistry that are involved and that are required to be known in order to venture into any medicinal chemistry or drug development topic. Okay. Thank you.